Hello, bio students. Today we are going to start Unit 6, and Unit 6 is called Classification. And we're starting with Lesson 1 today, which is Classification and Cladograms. So let's get started. So the word classify means to group things according to similar or different features or structures that they shared. Now, why do you think it's important to have some type of organization? So look at this grocery store, for example. What would happen if the grocery store would not be organized in the way that it's organized today? It would probably be very chaotic. So it's the same thing with species out there. There's over 13 billion species on Earth. New organisms are being found every day and identified, and scientists need to know where to put them. And when you understand a little bit more their classification, it also makes all these species that are being discovered every day, it also makes it a lot easier to study them if you are able to classify them in a particular way. So taxonomy is the scientific study of how living things are classified. So once an organism is classified, then we know a little bit more about that organism and its evolutionary history. So who started all this? What's in a name? So scientists, we know, are naming all of the organisms on Earth as they discover them. And it's funny because, like, if you get to discover an organism, then you get to name it, and you can pretty much select a species name that you would like. So just keep that in mind when you discover an organism. So this system of taxonomy was created by a Swedish scientist named Carolus Linnaeus, and this is his picture. He developed this system over 200 years ago, and he developed a system known as binomial nomenclature, and this is what his book looked like, Systema Naturae. And his book comes with some controversy. Maybe we can spend some time in class discussing that. But what he used is this nomenclature. Nomenclature means rules for naming organisms, and he used a binomial method, which means that you use two names. So what is binomial nomenclature, and how is it used to name something? So each species of a living thing is given a double name known as the Latin name or the scientific name. The first name represents the genus of that organism. So the genus tells which group of similar species the living thing belongs to. And the second name gives the species name. So it tells the name of the one particular species in that genus. So genus is a little bit more broad or broader than species. So why do we use this scientific name? So scientific name we know are universal among all countries, and we need to keep them universal amongst all countries because common names might differ depending on a particular region. And we know that common names are not very specific. Something might be named completely different in one particular region. So for example, this guy right here, do we call it a catamount? Do we call it a mountain lion? Do we call it a cougar? Do we call it a puma? Do we call it a panther? Well, what is it, right? So we all have to agree on a scientific name, and the scientific name of this organism here is the Puma concolor. And Puma is the genus, and concolor is the species name. So why is that important? Well, what's in a name, right? So we know that also languages differ. So we call these dogs if you speak English, but not if you speak French or Hebrew or Mandarin or Japanese or German or Hindi. So again, languages vary, so can a common name based on language, but not if we all agree to name that organism by its scientific name, which is Canis familiaris. So Canis is the genus, Familiaris is the species name. And we can all agree on that. So Linnaeus' system of scientific names gave different names to different organisms. So for example, a dog would be a Canis familiaris, a timber wolf would be a Canis lupus, and a coyote would be a Canis latrans. Now notice the word canis is used in all of these names. So this is the Latin word for a dog. So this tells us that a dog, a timber wolf, and a coyote are all in the same genus, right? Are all in the same genus Canis. So you can tell by looking at that they're all related in some way, right? Because they are in the same genus. They are all dog-like animals. Now the species is more specific because it gives you that particular type of dog, right? My little Mikey dog here has nothing to do with a timber wolf as far as the species name. Oops, lost my desk scribble. Gotta bring it back. Okay, so let's continue here. Okay, so there are some rules for binomial nomenclature. So these are the rules that govern binomial nomenclature. So the first letter of the first word is the genus name, and you always capitalize that. The first letter of the second word is the species name, and that is never capitalized. Now you have a choice. You can either use italics for both words, or you can underline the whole genus species. So this would be an example of Canis familiaris, right? So notice that the first letter here, and I've lost my desk scribble again. Notice how the first letter, goodness gracious, why is that? Okay, notice how the first letter of the genus here is capitalized, and notice how the species name is not capitalized, right? And then you can underline or put them in italics, and that's just proper nomenclature. So here's an example of a binomial nomenclature rule. So human would be a common name. Something wrong here with my desk scribble. Human would be the common name. Genus would be homo. Derived from the Latin word meaning hominid or man. And the species name for us is sapien, meaning wise. So boy, we're full of ourselves, right? We are a wise man. So that's the scientific name, Homo sapien. And of course, if we look at our evolutionary history and we look at our big brain, well, yes, right? We are very intelligent. We are very wise. If you look at our closest relatives, they did not make it, yet we did. So biologists classify all of the species on Earth. So if you're one of those biologists, then you are called a taxonomist, and you group organisms based on a particular taxon. And you can then study the phylogeny of the different organisms, meaning that you can look at the evolutionary past of those organisms. So taxonomists are constantly looking for clues to determining a species' phylogeny. How is that one species related to another species? So we know when we studied fossils in evolution that fossils provide a record of the unique characteristics of organisms over millions of years. And that's what that says here. You can't see it. It's word fossils, right? And fossils can be used to show evolutionary relationships. So we can radioactively date certain fossils, right? We can date the soil around the fossils, and we can see how old they are, and we can see how related they are. Here you see some of the phylogeny of our hominid species. So here we are, right? We are Homo sapiens, and you can put a picture of you right here. And then all of these other organisms are our closest relatives. So we see here that we are more closely related to Neanderthal man and Homo floresiensis than we are to, let's say, Ardipithecus ramidus that we know was discovered about 4.4 million years ago. And we are even less related to Sahelanthropus ticadensis man, which was discovered, or not discovered, but who lived around 6 million years ago. Okay, so we can also use embryos to help us determine the phylogeny of an organism. So we can tell that there are certain features that are present in certain embryos that are also present in other embryos of other species, and that's also going to help us understand the phylogeny of that organism. So here you see how perhaps a shark is not very closely related to a bird, right? But they do share some characteristics along the way, as you can see here. And we know that DNA is probably the most, or it's the strongest evidence for phylogeny or that evolutionary relationship. So we know that scientists now use DNA to classify organisms. So we look at the genome of a species, we look at the sequence of base pairs, and we try to see how closely related they are. So then we can use cladistics, which is how we organize these species, into how closely related the DNA is. So this is what a phylogenetic tree looks like. And phylogenetic trees are very similar to family trees, and they are based on the evolutionary past of an organism. So these lines here represent how related or how closely related they are, right? For example, you to a sibling, and then these right here would be your parents. And this would show how closely related you are to a cousin, right? And you are related because you share the same grandparents. And this will show how you are perhaps related to a distant relative, and that might be because you share some great-grandparents in common. Okay, so here you see that time is being represented by these branches of this tree. So the further down the tree, the longer ago the ancestor existed. So we know that our great-grandparents probably existed a while ago, right? So here you see cladistics, which is a tool that you use to classify organisms along their phylogeny. So here you see an example of a cladogram. So here is a cladogram, or a picture, that is used to organize these different species based on certain characteristics that the species might have. So notice down here, which is very important, at the bottom of the cladogram you have these derived characters. And these derived characters will show you certain characteristics that some of the organisms along this cladogram have. So for example, embryo retention in true plants. That means that that this characteristic evolved around this time period. So that means that algae do not have embryo retention. We know that vascular tissue evolved right here. So we know that mosses and algae do not have vascular tissue. So anything that falls under that particular derived character means that these organisms on the left side of the derived character do not possess those distinguishing characteristics, only those organisms that are on the right. So we know that only ferns and gymnosperms and angiosperms have vascular tissue because they fall on the right of the cladogram. If we look at seeds, for example, we know that the evolution of the seed came about here. So we know that ferns, mosses, and algae do not possess seeds because they fall on the left side of the cladogram. And we know that gymnosperms and angiosperms do possess seed. Same thing for flowers and fruit, right? So we know that the only organism that possesses flowers and fruits are the angiosperms because they happen to be on the right of this cladogram. Another thing that the cladogram can show you is the evolutionary timeline as far as appearance on this earth. So we know that these characteristics here are more primitive, so then that means that the organisms that possess them probably evolved on the planet a lot earlier than these organisms to the right. So then that means that the organisms to the left are more primitive and probably evolved first than the organisms on the right that are a bit more complex and probably have a shorter evolutionary history. Okay, so that's the definition of cladistics, a method of studying organisms, phylogenies, by determining relationships based on similar traits. So we looked at the cladogram in the slide before. It's a branching diagram that shows evolutionary relationships based on derived and ancestral characters. The closer two organisms are grouped together on a cladogram, the more closely related they are. And cladograms and other diagrams include clades, a group that includes an ancestor and all of its descendants. So here we see another example of a cladogram, and we see ourselves here. So being on the far right of the cladogram shows that we were, we just recently appeared on this planet, right? Our evolutionary history has not been as long as, for example, the ring-tailed lemur. So we look at some derived characters here, and we see that us as humans, we have all of these. We have 4 kinds of teeth, we have a large brain, omnivorous, all this. We have central eye area for more acute vision. We have a downward pointing nose. We lost our tail, and we have loss of opposable thumb on foot. So all these are characteristics that we've lost or that we either have or have lost, and that pertains to us humans. But if you compare that to a spider monkey, for example, right, the spider monkey, so the spider monkey does have central eye area for more acute vision. It does have 4 kinds of teeth, a movable head, and front-facing eyes. It has all of these derived characters on the left, but the spider monkey does not have any of these derived characters on the right. Okay, so if you were to again look at this cladogram, you will notice that we as humans, we are more closely related to the chimpanzee, right? Because we are closer here. We are closer on this clad right here than we are to the spider monkey or the rhesus monkey. Okay. So that's it for Lesson 1, Unit 6. Make sure you filled out your note taker. If you need to pause this video and re-watch certain parts, feel free to do so.